record because I forgot to do that. And make you big. And yes, Pearson will run slow, probably particularly on Sunday nights. And the reason for that is going to be because everyone in the country that's using Pearson that has things that are all due on Monday. Everyone's going to be on there trying to finish their homework or finish their quizzes or whatever. So this table, this is an answer to Colleen's question. This table in your book, and it looks like it actually give you the old page numbers up here at the top, I think, see where it says 84. So this is on page 85, I believe, of your textbook. So this is the important part of an amino acid that I want you to be able to recognize, that you can see this amine group, a carbon, and a carboxyl group. All of the groups in yellow, those are those R groups or those side chains, those functional groups. I don't expect you to recognize them and say, oh, this is phenylalanine. I need you to be able to look at this molecule and say, oh, that's an amino acid because it has the amine group, a carbon with a functional group attached to it, and then a carboxyl group. So this next picture, like this, right? So I need you to be able to recognize this is an amino acid. And if I put some of them together with peptide bonds, which I can't click on both of those at the same time. Let's see if I can make you really small. Yeah, there, I can make you go away. So this picture, when they start showing you this peptide bond, right? So when you put together a tripeptide, so that's 3 amino acids, you're not going to see that second oxygen or the hydrogen because that's going to become part of the water molecule that's part of the dehydration synthesis reaction to form this peptide bond. And the peptide bond, that particular word, this is a specific bond between the carbon of the carboxyl group and the nitrogen of the amino group of the next amino acid. This particular bond, this peptide bond, we're going to see that again later in the semester. So it is kind of important to remember how they are joined together. And all of these, when you build polymers, are going to be by dehydration synthesis, water coming out, right? Hydrolysis, to break that bond, you have to put water back in so that one of the hydrogens can join this amino acid and the hydroxyl group can join the carboxyl of the previous amino acid. Usually what people have more trouble with is this part, the function of proteins as far as they're two-dimensional, this picture here, the two secondary structure, tertiary structure, quaternary structure. So I would watch, there's 4 levels of protein organization, and I would watch these videos. I don't think, my little laptop's gotten a little hot here from having so many windows open. I don't think I can show you this video directly, but I would recommend that you watch it if you haven't already. Because for proteins, structure dictates the function, right? So the primary structure is just the chain of amino acids. The secondary structure comes in these 2 forms. And then, oh, I've got quite a picture here. This is the tertiary structure, and that tertiary structure is going to be particularly held together with various chemical bonds. And then the quaternary structure, when a protein has multiple subunits. Many, many proteins, unless they are peptide hormones, which you won't meet again until anatomy and physiology, have this at least secondary structure, and most functional proteins have quaternary structure. This is the poster child, hemoglobin. And again, I would watch these videos. And I thought of something else I wanted to tell you about proteins. So probably further back, they show you the functions of proteins. I think they open with that if they're keeping the textbook the same. This panel here. I don't expect you to be able to tell me examples of all of these, right? But I would expect you to be able to recognize the list. And if I put something on the list that doesn't match this list, that you would say, oh, that's not a protein, a function of proteins, right? So this little video is actually quite good that goes through the different, showing you inside a cell the different functions of proteins. And I realize this makes less sense now because you don't know that much about cells yet. It will make more sense later after we start going through cells and getting things a little more complicated. Trans fats, specifically, probably not, Angelina. No. Chemistry, go to chemistry, learn all about it, right? These chapters you'll find repeat in your next courses, in your 200 level science classes. So even if it's the plant science class, if it's microbiology, if it's anatomy and phys, even if it's the environmental class, this chemistry stuff just keeps repeating. And those of you that took bio late in high school, you'll recognize it all again. You're just like, oh my gosh, organic molecules again. Because and then it just keeps getting to be a smaller and smaller portion of the intro of the textbooks. So what's going to happen is your next textbook is going to compress everything from chapters 2, 3, 4, and 5 into one chapter and say, review this because this is the chemistry of biology. These are things you need to remember. So a lot of this stuff we go through a little more slowly in Bio 1. And then
No, she just started it.
No, yeah.
Hey.
Good morning. So a lot of this will just keep showing up over and over again in smaller and smaller sections, but you have to remember it. So kind of why we slog through it in Bio 1, which is most unpleasant, but necessary.
I have a question.
Yep.
I know we talked about it a little bit, but are we going to have to find the pH based on that log equation for the test? Or
no, you just need to be able to recognize that that's the equation, that's the actual calculation for pH. Okay, so like if I put up 4 different equations, you just need to be able to say, oh no, pH is the inverse log of the concentration of the hydronium ion. That's what you need to be able to, and you just have to be able to recognize it in Bio 1. Again, it's chemistry, and I realize some of you are taking chem at the same time, so it makes more sense to you. pH is such an important thing for cells and for living things that we have to go through it in a little bit more detail than I would really think is important for Bio 1, right? I'm not going to ask you specific things if they're acids or bases unless I tell you what their pH is, right? You should know that any number less than 7 is an acid and anything more than 7 is a base. You should know the steps on the pH scale are in the power of 10. It's a log scale. That's really important. And you should have an understanding of what a buffer is in cells and how that can help a cell or an organism moderate pH changes in the environment. Right? All right. Other questions, comments, worries about the pending doom? It'll open.
Yeah, I got a question.
Yes.
I know that we didn't go over this in my lab in mastering. Are we going to go over diffusion, osmosis, facilitated diffusion, and active transport?
So, Michael, those belong in chapter 7.
Okay.
And that's not on this exam. So
All right.
This exam is just chapters 1 through 5. So those videos I don't think are even, I don't know if I even posted them in the next module yet or not, because those go with 6 and 7. Did you see them in some of the videos about that protein function video, or are you just wondering?
I'm just wondering for the future. That's all I'm doing.
Okay, so you have to look at the note outlines, right? And if you look at the note outlines, there's nothing about any of those topics on the note outlines from chapters 1 through 5. And there's nothing, I don't think there's anything in the videos that are posted 1 through 5 about that particular topic.
Okay.
Do we need to know anything about asexual reproduction?
You should know that that's from the list of things that are prokaryotes and eukaryotes. Probably, you should be able to tell me definition of asexual reproduction, yeah. So if I were to ask you characteristics of living things, and you listed reproduction as one of those characteristics of living things, I would expect you to tell me something about it other than just say reproduction, right?
All the questions on the test are multiple choice, right?
I think so.
And how much time do we have for the test?
You'll have 45 seconds per question,
Okay.
unless you have an accommodation.
Well, how many questions are there?
I have no idea. I thought, sorry, I thought I was gonna look at it yesterday and then I didn't. So, I think, remembering from summer, I think there's probably, it should take you about an hour, maybe. I think it's in that range. Because I realize for most of you, you can't just sit down and dedicate more than that amount of time at once. If we were live in person, we'd have a 2-hour time block or an hour and 50-minute time block and a lab period that we would take an exam. So that's, and usually that first exam is 1 through 7. So I've shortened it to 1 through 5 to try to keep it within, I think it's an hour or less. Now it's recorded. Okay, so I'll double check that, but I think it's an hour or less. So the chapter touched on just a question from Colleen, messenger RNA and transfers specifics. No, not yet, not until we get there. I think so. I think, Samira, you get the scores right away. I know there's a couple questions that I have to check and make sure the right answer is selected, but I think you get them right after you finish the test, yeah. So you know right away. And if you've done all your homeworks up until now, it'll show you if you look in Canvas, like I don't block that either, it'll show you your cumulative, like total percentage so far as it goes along. I know some people turn that feature off. I don't care. I think it's important for you to know where you are as we go through the semester. So those are the elements that are important. So S is for sulfur, C for carbon, N for hydrogen, nitrogen, oxygen, and phosphorus. Right. So if you watch, you got to watch those videos, guys. I know they're boring, but if you watch them, it just goes exactly through the notes. And I draw on the board or I show you the pictures right from the book that are related to those. So I get it. You look at the note outline, you're like, oh, this is easy. I just have to fill this in and I'll be ready for the test. I would watch the lectures because that's like if you were in class, that's going through them. Really? That's weird because I thought it specifically put that there. So your book lists maybe 2 others. I think they list calcium and sodium and chlorine or something in that list. But it's probably also in that same part of chapter 2 where it talks about the elements that show up in the highest concentration in living things. That's the list. I don't think they've actually released, I still can't get my hands on a copy of the completed textbook, but I'm just gonna flip through my book real fast. I think there's so in the, whatever edition this is, the 11th edition, see this table, you probably can't see it, but there's a little table. That's what it refers to, whatever page that's on. And you'll notice that oxygen, carbon, hydrogen, nitrogen make up 96% of your body mass. And then all the rest of them only make up 3.7%. But I picked out some of the ones that I thought were, that show up a lot in our discussions of chemistry later on. Like the sulfurs will show up because of disulfide bonds and proteins. And I put the phosphate on there because obviously because of ATP. All right, anything else? So that's a good question, Colleen, because everyone needs to hear it again. So the first exam will be, it'll open tonight at, well, it'll be tomorrow morning at 12:01 a.m. and it'll be open all for 24 hours. It'll be open until tomorrow night at 11:59 p.m. So it'll show up in the module. Right now it's unpublished. So I thought you could see the list, but you couldn't actually click on it. But I don't know. Go back to my Canvas page here. Close the book. So let me share that screen with you. So here, can you guys see the screen? So here at the end of this module 2 here that has chapters 2 and 3 in it. And then here's the one with chapter 5 in it. Down here at the bottom, oh, I see, that's why you guys were asking about 6 and 7. But because I want you to read that ahead so that on Wednesday we can talk about it. Here's the exam. So maybe you guys can't see it yet. If I click publish, then it'll be, and it has a due date on it right now from the summer. So it'll be sitting right here at the very bottom of this module, of module 3. And I'll put that for this first test. I'll be sure to put that in an announcement. Right, I'll probably forget after that, but you should, I also leave my quizzes in Canvas. So see how in your toolbar here, see how quizzes is available? You can see all these other ones, right? But they're closed. You can't get to them because they're from summer. I guess I can hit publish. Right, so now you can see lecture 1. So you can get them, you can get to the quizzes or the exams through quizzes or through the modules. And in the modules, they'll always be the last thing in that module. So like right here, it sits at the bottom right there. So I am going to publish that one right now so that and then it'll be live starting, like I said, it'll open up at 12:01 a.m. tomorrow. So just after midnight tomorrow. And then it will be open all the way, just after midnight tonight. It'll be open all the way till 11:59 tomorrow. Don't wait until 10:30 or 11:30 to sit down and take the quiz, because there's 80 of you, and if everyone's trying to get into that exact same quiz all at the same time, you're probably going to have trouble, right? I mean, I would expect technical difficulties. So I would take it when you're ready, but I would try to take it and take it on a computer. Don't try to take it on your phone, and be someplace where you have good internet connection and be someplace where it's quiet so you can concentrate, and it'll probably take you about an hour, like less than an hour. So Allison, you don't need to know the different shapes of the orbitals and the different names of the orbitals, but I would expect you to be able to tell me that the first electron shell holds 2 and the second one holds 8 and the third one holds 8. But you don't have to be able to tell me what the shape of those orbitals are, like the ones that are all like dumbbell shaped and stuff like that. No, chemistry, more chemistry than you need to know. So just so you know, I don't know how many of you guys have experience with Canvas. Um, it will indicate to me when you've left the screen. It also indicates to me how long you take on each question. So please be mindful. We expect this to be taken like a regular exam where you sit down and just take the test. There is not time for you to look up all the answers. You shouldn't be sitting there trying to look up all the answers. It's an exam. It's not a take-home. It's not a homework assignment. Okay, so I know that works differently in different courses depending on your instructor, but at least in Bio and Bio1, we've tried to set them up so that they're like as if you were going into class and sitting down to take a test. So if you're not prepared, you're not going to get finished. So you need to study ahead of time and be ready to answer the questions. Best advice I can give you. I know it's stressful. I know you feel like it's very nerve-wracking because you're like, oh, I only have 45 seconds. I can't. Just focus on, if you're ready, you can do it. If you were in a real classroom and I gave you that amount of time to take a test, you could do it. So you can come back to a question, Olivia, but they're only going to be displayed one at a time. So you might run into, so you can come back to a question, but be careful how many you save for the end, because you'll have to scroll back and try to figure out which ones they were and where they are and how many there were. So I think so. Canvas is pretty liberal with those kinds of things. I don't think we can, I don't know. I think I can block you from doing that, but I feel like if you're taking a test and you don't know the answer to something, there's no point in you wasting your time on that question. You go on to the next question, and then you should be able to go back to it. So I think that's how I have it set. You can go back. We'll have lots of tests or quizzes, like 9 of them, so that you have lots of chances to improve your grade. Remember, the quiz portion is 70% of your grade, and the labs and the mastering add up to 30%. Right, so Olivia, the 45 seconds isn't a timed thing for each question. It's a timed thing. That's how it's determined the length of the test overall, right? So all I set is that you have 45 seconds for each question. And that just dictates how long Canvas is going to let you be in to answer all the questions. Does that make sense? So right, if a particular question, 45 seconds is pretty standard between 30 and 45, 30 and 50 seconds. Most, you should in college be able to read a question in about 3 seconds, process it in the next 10 to 15, and then make a decision. So you should have plenty of time to answer each question. Yes.
How reliable do you think like the Amoeba Sister videos are? Because I like to look at those because the pictures and stuff kind of help me remember certain aspects. Do you think those are like reliable enough to look at?
I think they're great to look at. I think they don't have enough detail in them.
Okay.
So I think they're great to look at early on. And if it helps you remember like a particular, right, if it helps you put a picture in your mind for a particular item. But I've found the few that I've watched, I would say high school-ish level, right? And so we're trying to get you to the next level. And so we do expect that you have a little bit more of the details. And I found them kind of variable. Like some of them I'm like, oh, that's everything I would ask. And then other ones I'm like, so I would watch them early in your studying, and I wouldn't use them for the end and memorizing things, but I would use them early to help get you oriented. Absolutely.
Okay, thank you.
Welcome. Everybody likes the Amoeba Sisters. All right, other questions you guys have? Comments, worries, concerns? Well, you have from now till midnight to try to cram everything into your brain and then take the test, or partly tomorrow morning or tomorrow afternoon. But yeah, it will close at 11:59, and then if you don't take it within the time, you'll have to take a makeup test, and those are, I schedule those as oral exams, and we just meet as a private Zoom meeting, and I just ask you questions. So those are much worse, which is the point, to make you sit down and take the test. That's a little bit easier. Email me today. I'll definitely be on. I have office hours from 9 to 11. I'll be on my email and on the computer definitely between then. I have a Zoom meeting with my micro classes from 11 to 12. And then I'm usually not on very much in the afternoon because I feel like I've spent 5 or 6 hours working on the computer already. But I will check my email again. I'll check it very frequently through evening and tomorrow morning, obviously while you're getting ready to take the test. So if you have a 911 where you're just like, I don't know how to do this, or I don't know the answer to this in the homework or whatever, just let me know. Alrighty then, I'm gonna stop the recording.